Hi, my name is Jordan Whitlock, and I'm here to talk to you guys today about the role of cell-specific expression and regulation in Mendelian disease. What's really interesting is that individually rare diseases only affect a small proportion of our population, about approximately 25 million Americans. However, there today exist almost 10,000 Mendelian diseases with 300 new Mendelian disease genes discovered each year. Mendelian diseases are caused by germline aberrations that occur early on during fertilization between the sperm and the egg. Because of this, the resultant variant is incorporated into all daughter cells and tissues so that the entire organism theoretically carries that variant throughout. What's really interesting is that when we look at an online database referred to as the Online Mendelian Inheritance of Man, or OMIM, for all of the 10,000 known Mendelian diseases, for 1/3 of them, it's unknown what the molecular variation is that causes the disease. For 2/3 where we do know the molecular variation, so that might be the gene or the causal variant associated with a phenotype, for half of those cases, it's unclear to us how that actual causal gene or variant leads to the disease. And so this kind of serves as the basis of our research project and focus. And there are some different hypothesized mechanisms underlying that proportion of diseases where we don't know the mechanism leading to the phenotypic manifestation of the disease. And so if you look here on the screen in the bottom left, we'll see all of the different hypothesized mechanisms of tissue specificity that exist today. They range across multiple different biological processes, such as epigenetic-based mechanisms in red, genetic-based mechanisms in teal, expression-based mechanisms in purple, tissue-disrupted network-associated mechanisms shown in pink, non-cell autonomous or epigenetic mechanisms in orange, and then regulatory-related mechanisms in green. And so for our research in our lab, we focus on both expression-based mechanisms of tissue specificity of disease and regulatory network-based mechanisms of tissue specificity of disease, but focus on, instead of the tissue level, the cellular level, because as we know, a lot of the underlying cellular heterogeneity is lost or masked in bulk approaches. And so using 3 different multi-omic input data types, protein-protein interaction data from string, single nuclei RNA expression, sequencing data from publicly available databases for human and mouse, as well as data that we have generated ourselves, and transcription factor motif information from the online database referred to as SysBP, we take those 3 inputs and construct cell-specific regulatory networks using a regulatory inference approach referred to as PANDA. And so PANDA stands for Passing Attributes Between Networks for Data Assimilation. And what this allows us to do is to take the different protein and motif and expression inputs and construct cell-specific regulatory networks showing relationships between transcription factors and genes, so that then we can analyze and compare the differences in these cell-specific networks across different gene sets of interest. And so for us, that would be anything that is renal and neurological associated, so linked to a phenotype that presents in the brain or kidney. And we took and compiled a list of genes from different existing monogenic diagnostic disease panels, and then used the API key to access and query the OMIM database, which contains those genotype-phenotype relationships between genes and known phenotypes within humans, to build a gene set of over 1,000 different renal and neurological associated disease genes. And so once we've constructed these cell-specific regulatory networks, we can then go in and pull out subnetworks for all of the disease genes associated with ciliopathies or developmental delay or epilepsy or polycystic kidney disease, any of those Mendelian diseases associated with a brain or kidney phenotype that are of interest. And then we can take these subnetworks that contain those disease gene of interest or our favorite disease gene of interest and look at the different transcription factor activator and repressor roles of those Mendelian disease genes across the subnetworks between cell types, detect and see if there are special communities of transcription factors and genes interacting in one cell type compared to another, perform differential edge analysis, and then see what pathways are differentially regulated across the different cell types within the brain and the kidney. And so in doing this, we started our research by focusing on a gene called SETBP1, which is known as a DNA master regulator and transcription factor within the body. And it's associated with 2 different Mendelian disorders, Schinzel-Gideon syndrome and SETBP1. However, it's unclear what the actual mechanism that leads to each of these disorders to present with either a phenotype within the brain, such as neurodevelopmental delay, or within the kidneys with renal dysfunction. And so we're trying to, using these methodology approaches, not just understand some of the basic cell-specific regulatory drivers underlying Mendelian diseases so that we can understand what may be causing the phenotypes we see in clinic, but also using SETBP1 as an example there, because we have obtained different rare disease mice containing a point mutation in SETBP1. And so one of our goals is to understand how a variant may influence cell-specific regulation across different cell types that we can link back to the overarching phenotype of disease, which has been shown in the literature with examples of cystic fibrosis and rare pulmonary ionocytes contributing to overarching disease phenotypes within mice and humans, as well as with type 2 diabetes, where cell-specific regulatory communities are linked back to the overarching phenotype driving type 2 diabetes. And so we have investigated some of the cell-specific relationships for SETBP1 as shown on the screen across different prefrontal cortex cell types from publicly available human data. And we see that when looking at the edge weights, which just represent a relationship between a transcription factor and a gene and how confident we are that there's a regulatory relationship present, we see that there is differential regulatory edge weights across the different cell types. And looking at subnetwork containing SETBP1, some of the SETBP1 astrocyte-specific relationships are highlighted in purple. And so we are excited to, in the future, take these cell-specific networks using the data that we've generated ourselves, which just came out of the sequencer this week, and then look for not just subnetwork differences for Mendelian disease groupings or renal and neurological associated diseases, but also within SETBP1, and compare some of our findings in the mouse back to the human. And I just want to thank the Lassane Lab and the UAB CMDB T32 Fellowship for funding.